Good morning, everybody. We'll get started in just a moment. I want to welcome everybody, and so glad so many folks are able to join us for a topic on soil health today. So, in the spirit of soil, if you can, in the Q&A box, or actually in the chat box, if you can put down what you think our Florida state soil is, and enter that in the chat box if you can, and then we'll get started in just a moment. A lot of people saying sand. Deirdre, good job. Angela, good. You got it as well. I bet a lot of people didn't realize that Florida has an official state soil. Jennifer, very nice. And Shelley too. Carol, good. Well, fantastic. It looks like most people it's slowing down the number of people entering. So I think we'll go ahead and get started. Good morning. I'm Tom Wickman, the Assistant Director of the Florida Friendly Landscaping Program, and I'd like to welcome you to the Florida Friendly Landscaping Professional Webinar Series. Let me start by saying that I hope that everyone that was affected by Hurricane Helene is getting the help that they need, and getting things cleaned up and taken care of. Certainly that was a devastating storm for so many folks. With that being said, we have Hurricane Milton approaching and looming off the coast, and so we want to make sure that everybody does what they can to make themselves safe, and protect their property and their families. And we here at the Florida Friendly Landscaping Program certainly uh, you know wish you the best, and hopefully everyone makes it through with little to no damage. But do take those steps that you can to get help, and any steps that you need to try and make your property as safe as possible. I know there's a lot of people that won't be able to join us live today because they're busy prepping for the hurricane, but I do want to mention to everybody remind you that all of these webinars do get posted to our website, to the Florida Friendly Landscaping website. You can always see all of the archived professional as well as homeowner webinar series. They're there. You've got 4 years of webinars. So if ever there's a day that's not good weather, or you just have the opportunity to learn something, go to the Florida Friendly Landscaping website at floridafriendlylandscaping.com, and you can access all of those archived webinars. So let's talk about today's webinar. The topic today is taking the temperature of soil health. And today we're going to have Dr. Erhunmwunse talk about this topic, and it's such an important topic that we don't often deal with, and that's the first time in the last 4 years that we've really touched on soil. So I'm really excited about today's presentation. Your microphones have been muted. We will be closing the chat feature. By the way, I would like to address that, and I was so happy to see so many people get the fact that our official state soil is Miaca fine sand. A lot of people put sand, and you're not incorrect. It's just not quite there yet. It's Miaca fine sand is our official state soil. And so we will be closing the chat feature. Any questions that you have, do add those to the Q&A box, and we'll take those at the end of the presentation. This webinar is approved for 1 FFLCP, LIF, FDAX, LF, LCLM, LLO, CLO, and ONT, or DBPR, FWS, and 1 FNGLA CEU. There is a $15 administration fee to receive a certificate for continuing education. We'll put a link in the chat box to make payment for the certificate if you've not already done so. We'll submit the CEUs to the licensing agencies on Friday, so make sure if you want your CEU to make payment by the end of the day today. This is part of a monthly webinar series held on the second Tuesday of the month at 10 a.m. Eastern Time each and every month. Our next webinar will be held on November 12th and will be on Palms of Florida with Stephen Brown. Also, you'll see a survey invitation pop up. Please take a moment to fill this out. It's really important that we get that filled out. This is about the time of year that we really start planning in earnest for next year's webinars. And so we want to know what you want to hear and those topics that you're passionate about. So it really helps us choose our programming. And with that, I will introduce our speaker. Today, we're so fortunate to have Dr. Erhunmwunse, who's from Edo State, Nigeria. She received her PhD from the Department of Soil and Water and Ecosystem Sciences at the University of Florida. She's currently working as a postdoc associate with Dr. Sarah Strauss in Southwest Florida Research and Education Center in Immokalee. There, they investigate the impacts of management practices and agricultural systems on soil health and soil microbial communities. So with that, I would like to turn the floor over to Dr. Erhunmwunse. Go ahead and take it away.
Okay, thank you so much for the introduction. And thank you all for joining in today. So today's topic, like he introduced, the temperature of soil health. When we talk about the temperature of soil health, I would like to compare that to humans. When someone says, oh, you're not feeling fine, sometimes what we'll do is we'll start from the temperature and other physicals, right? You take the temperature of the person, oh, is it running at 100 degrees? Is it running at 120? So what are we doing? We're monitoring the individual's health. It's the same thing with the soil health. We use the word temperature, and that is we comparing and saying monitoring soil health. Before we go into how we monitor the soil health and talk about that in details, it's a good thing to first start with, what is soil health? So my presentation today, I will break it down. I will start by introducing to you and telling what soil health is really about. And then we'll look at the key indicators for monitoring soil health. And then we'll also look at various management practices that we have done in our research lab and other research labs and how it affects soil health. We'll liken that to Florida because we do have unique soil properties when we compare to other regions, especially in the temperate region. So with that, I will start by introducing my name. I know it's really, really long, but the truth is, if you just take out a lot of the consonants, it's easy to pronounce. So my name is Adesuwa Erhunmwunse. What is soil health? There are a lot of definitions for soil health, but the one that I used today for this presentation is the one adapted from USDA, Natural Resource Conservation Service. It defines soil health as the continued capacity of a soil to function as a vital living ecosystem that sustains plants, animals, humans, and also the environment. When we talk about humans, we're talking about the environment. You want the air to be clean. We want the water to be good for recreation, to be good for drinking, and all of those things are tied to soil health. Now, the unique thing about soil health is it's different from other terms that we have seen in the past. Maybe we're familiar with them. Talk about soil quality, soil fertility, And now there's a new one that is common, a regenerative agriculture. But the beautiful thing about soil health is it includes soil biota, especially the microbes. So when you talk about looking at the soil as a living ecosystem, it means we have to consider everything, a comprehensive assessment of the soil. And that includes the soil microbes. And we know they are living. So considering that, let's move forward. The soil health, we try to create an analogy, like I said before, to human health. For the human health, we can loosely categorize it into two categories, the physical aspect, right? Or we talk about you have to eat the right diet, you try to exercise, don't sit for too long. All of those factors affect the human health. And then we'll talk about the genetics, that the innate structure of the man, what we're made of, and that we cannot control. That is similar to the soil. The soil, we're talking about the innate characteristics of the soil. Where is the soil from? Where does it originate from? The parent materials that the soil is formed from, that would affect the soil and the soil types that we have. You see, for example, we have in Florida, we have the spodosol as our national soil. It's sandy, that's because of the parent materials, the origin of the soil. That we cannot control. But the other aspects of the soil that we can control, the management practices, in this case, that's the exercise for humans, then the diet, what do we put in the soil, the type of fertilizer, the type of, is it organic, is it inorganic, how does it affect the accumulation of nutrients in the soil? So that aspect, these are the different factors that would affect soil health in general. And measuring the human health, these are the various factors we'll look at for humans, right? We'll look at the temperature, blood pressure. When one visits a doctor, they are going to look at these things first before they start diagnosis of the individual's health. Temperature, we'll look at the blood pressure, the weight, cholesterol, and many other health indicators. And that is also similar with the soil. When we start talking about the soil health, what are the indicators we look at to really diagnose the health of the soil? We're going to look at the texture, we'll look at the bulk density, we'll look at the soil organic matter, the pH, the nutrient availability for that soil. Then we'll look at the microbial properties too. Enzyme activity, soil respiration, these are the indicators. We call them the soil health indicators that we measure to look at the soil health. Now, like humans, not a single set of numbers can you use to monitor the health of a human, right? For example, the parameters for measuring a healthy 65-year-old is different from the parameters to measure a 2-year-old. That is also similar with the soil. There is not a single parameter or a single key indicators for measuring a healthy soil. For example, the soil health indicators for the Midwest soybean is different from that of the Florida tomato system. So that tells us that soil health is context dependent. And that is why for us to say, oh, the soil health indicators for this system, we have to carry out studies for that system to be able to make, to draw conclusions on a particular system. Back again to soil health indicators. We have different indicators. We have the physical indicators, which are the ones listed here. You're talking about the bulk density, the soil structure, aggregate stability. How does when you grow and plant a particular plant, let's say vegetables in a particular soil, and the water you're either maybe using a rain-fed system or irrigation, does the water get to the plant? Is it that the soil is still, when the soil is clayey, for example, that would affect water infiltration in that system. So those are the physical indicators for measuring soil health. And then we'll come to the chemical indicators. These will look at soil pH, nutrient availability. Does that plant in that soil have enough nitrogen? Does it have enough phosphorus, potassium? Is it available? And then we also look at the cation exchange capacity. Is the plant able to get these different cations that becomes nutrient needed from the soil, or does the soil just suck it in and refuse to release it to the plant? Then we also look at the biological indicators. Look at the microbial biomass, the soil enzymes, the earthworms, particulate organic matter. Then we look at soil microbial communities, the bacteria, fungi, diversity, the community composition, which is what our lab focuses on. And that's what I'm going to be talking about in details today in this webinar. Now, putting it nicely into a graph, I wanted you to see a combination of how the different indicators that I talked about, the soil physical, soil chemical, biological, how they interact. And then we have this, you see, when the soil physical indicators and soil chemical indicators interact, we call that physical chemical properties of the soil. But when you have the interaction of the three, soil physical, soil chemical, soil biological, then we have the term that we are talking about today, soil health. And now you see this big figure here. Different factors are affecting these different indicators, which definitely would affect soil health. Your climate, type of rainfall. Are you getting rainfall consistently? With Florida and the subtropical climate, temperature, it's good enough. So we're getting active decomposition, which makes soil organic matter low in the system, coupled with the parent material where the soils in Florida are formed from. And then we talked about management practices. What type of crops do we grow? What animals grazing? What are the other practices, tillage in the system? All of these factors would affect different key indicators and in turn affect soil health. But the beautiful thing, like I said, about soil health, when we talk about it as a living ecosystem, it means that the microbes are critical to most of these indicators. And that is why it's very important to study them. And that is what our lab does. So most times I was opportune to carry out on-farm studies with growers, with ranch owners and growers in across Florida, North Florida, Central Florida, and South Florida. And most times when I interact with them and I talk about microbes, most times what comes to their mind is, oh, they're really bad. They affect our crops. They affect our plants. But now my question to you today is, what comes to your mind when you hear microbes? Do you have the same thought process as the growers that I worked with? Do you think of pathogens? Now you see the citrus here. You see the leaves are affected. They're going bad because of the popular HLB disease, which we also know as citrus greening. Is that the same thing you think of when you think of microbes? Is it just pathogens? But I'm here to tell you today that we have beneficial microbes and they do great things in the soil. Before we go into why soil microbes are important, I just wanted to paint this picture of how gigantic the microbes are in the soil. If you get 1 gram of soil, which is equivalent to 1 teaspoon, you get more than 1 billion microbes in that quantity of soil. They are a lot. And inside that 1 gram of soil, you get we have 500,000 different species of bacteria. So having this knowledge today, you can see why it's okay to think of soil microbes or microbes holistically, not just the pathogens, but also looking at the beneficial ones and how we can help stimulate them in soil so that they can keep doing the good things that they do for the soil, the environment, and our plants. Now, we have different ways of studying these soil microbes. We have the cultural dependent method, whereby we take agar, or we'll call them media, food for the microbes. We put them in the plates in the lab, and then we're able to culture these microbes. But the thing about this method is that only 1% of soil microbes can be cultured. So it means if we're using just these techniques to study microbes, it's not enough to cover the diversity of soil microbes. And the good thing about this is that there is an advent of another method where we can use to study the 99% of soil microbes that cannot be cultured. And that's where this figure comes to play. Now, these are the microbiome. This is a soil profile in the soil. And we have different methods to study them. And this method is the previous one I just showed, the culture dependence method. And if it's just helping us get 1% or study 1% of soil microbes, it means that the diversity are being underestimated. But when you go down and you see this other method, which we call culture independent method, which is what we use a lot in our lab, we are able to estimate the different microbial diversities in soil and other environmental systems. And this method is just like what I said earlier when I said genetics, humans' genetics, which affects the soil. The microbes too, we're looking at the genetic materials and using their genetic materials to study them, whereby we look at the microbiome composition, their functions. We use we call them gene markers. There are different techniques being used. You have the metagenomics, amplicon sequencing, taking the genetic materials of these microbes and sequencing them, using that to identify who they are, what they do, and how they are connected. And then we use software such as Chime, Mother, Arrow, and Python to analyze our results. And then from that result, we're able to make inferences of how different management practices affect soil microbiomes and how also it affects their potential functions and their relationship. That way, we're able to answer a lot of questions just using the culture independent method. Now, with this method that we described, what are we basically looking at? We're looking at who is in there. I'm talking about the soil microbes in this case. Who are the microbes there? And the next question we ask is, what do they do? Are they helping us improve the aggregation of our soil? Are they helping us release nutrients? These are the questions we ask when we do the culture independent method and even culture dependent method to study soil microbes. And the last question we ask is, how are they connected? It's just like we humans, we have families, we have brothers, we have sisters, we have extended relatives. So there's that connection when we have the family tree. It's the same thing with microbes. They have that phylogenetic tree that shows the relationship. And that is what we do in Dr. Sarah Strauss' lab, soil microbiology lab. We use this different method to study these aspects of soil microbes. Now, back to my question. When you hear about microbes, what do you think of? Do you just think of pathogens? If that is your thought process, when I go through the next few slides, I think that perspective will be changing today because you're going to see the cool stuff that soil microbes do. First of all, you have those plants here where they are above ground, below ground. The soil microbes are below ground. They also affect the above ground, but in soil microbiology lab at Southwest Florida Research and Education Center, we mostly focus on soil microbes below ground, the roots, rhizosphere, and also the bulk soil. These microbes are involved in soil organic matter formation. Yes, when we release organic materials into the soil, they are the ones involved in decomposition. They are the ones involved in mineralization, in which all of these processes lead to soil organic matter formation. And they also, when we go down, we see that they are also involved in disease suppression. How do they do that? You see some of these antibiotics that we use, some of them are formed from microbes, or we've seen how they form them, and then they go about going about developing it just based on the pathways that microbes use in the formation. So that means that even in soil or in plants, these microbes are secreting these antimicrobial compounds, which to them is a secondary metabolite. And when they release these compounds, they help in suppressing pathogenic microbes in the system. So that tells us the importance of soil microbes. Apart from that, they are also involved in root growth extension. They can do that physically or through hormone secretion. I'm sure we've heard of the popular mycorrhizae. The hyphae for mycorrhizae, the diameter is very small compared to the plant roots. So when the mycorrhizae fungi forms a symbiotic relationship with plant roots, it means that they are extending plant roots physically. And that way, they are able to access soils and regions that the plant roots cannot access. It increases the nutrients. It increases the area of soils that they can access and take up nutrients and transfer it to plants. And the plants in return will give them carbon. So that's a win-win too for the plants, because that way the mycorrhizae fungi, they will help take up nutrients that the plants do not have access to. And not just nutrients, they also help in water uptake. Apart from the root growth, we can still see that the microbes are also involved in nutrient cycling, nutrient availability. We'll talk more about that in the next few slides. Now, this is soil microbes and soil organic matter. See what we'll talk about. You add organic matter in the form of maybe plant litters or in the form of compost or other forms of organic material. This would stimulate biological activities, soil microbial diversity, which also leads to decomposition of the organic materials that have been added to the system. And as a result of that, nutrients have been released to the plant. Plant growth promoting substances like hormones that I said earlier, they have been released to the plants. And these microbes in return too can also detoxify harmful substances that can affect plant growth. In addition to that, due to decomposition, they help in aggregation stability, pore structure improved, whereby water, air, aeration is improved, water infiltration into the soil, which also the plants will take up, is improved. And the increase in soil microbial diversity and activity would also reduce soil-borne diseases and parasitic nematodes. So microbes are very important in soil and helps in plant growth promotion. Now let's talk about the microbes and nitrogen cycling. Nitrogen cycling is just taken out of the numerous nutrients in soil. You see, the popular one is biological nitrogen fixation. When you see legumes, which I will talk more about because I do have a slide on it, you see nitrogen in the atmosphere. We have 78% of nitrogen in the atmosphere, but the plants cannot take it up because it is in the form that plants cannot use. But when plants form symbiotic or mutualistic relationship with soil microbes like rhizobia, it helps to take up that nitrogen in the atmosphere that is not available to plants. The microbes will take it up and break it into forms that plants can use. In the process, we call nitrogen fixation. It breaks it because the plants take up nitrogen in the form of nitrates, ammonia. And that the microbes are helping to convert the atmospheric inert nitrogen into forms that plants can use. And the plant is able to take up that nitrogen and use it for growth. In return, it gives carbon to the rhizobia and also shelter to the rhizobia. In addition to that, you also see nitrification, mineralization, organic nitrogen in the system, maybe through organic residues, is being mineralized into ammonium forms, ammonium ions that plants can use. And these are just the ability of soil microbes in nutrient cycling in soil system. And then we have nitrification, we have denitrification. Soil microbes are responsible for all of that. See how important they are in the soil systems. Apart from nutrient cycling and soil organic matter formation, soil microbes are also involved in promoting plant health. And how do they do that? We have plant growth promoting bacteria as well as fungi that can assist with nutrient acquisition for plants. I already gave an example of mycorrhizae. The hyphae are so tiny, with tiny diameter. So it means that the places that plant roots cannot go to in the soil, these hyphae can access those regions. And it increases surface area for the plant roots, which means more water, more nutrients. Apart from that, these plant growth promoting microbes also, they release secondary metabolites like hormones that would help stimulate plant root growth. They also defend against pathogens, which I talked about, antimicrobial compounds being released. They also lead to aggregation of soil. Especially look at the Florida soil, it's so sandy, the Spodo soils, Altisoil, Antisoil, so sandy that even when you're trying to get your soil cores into the sand, it doesn't just form, it just breaks apart. And that's because of the low aggregate stability of the soil. But when we carry out different management practices to improve soil health and soil microbes, these are all of these factors that will try to help improve in the soil so that the plants would have a healthy environment and also healthy growth. And the way we look at these soil microbes, you're looking at the roots, you have the rhizosphere, you have the rhizoplane, and you have the endosphere inside the roots. And it's just looking at the relationship between the roots and its association with the soil. The rhizosphere is outside, very close to the bulk soil, and then the rhizoplane is looking at the roots inside, it's next to the rhizosphere, then the endosphere is just inside the roots. So when we study soil health, we also look at the rhizosphere, we look at the bulk soil just to see how whatever management practice we introduce, how it affects soil microbes and its impact on soil health. See on soil microbes and plant health. I already talked about rhizobia, symbiotic nitrogen fixation. Now look at it. For man to make nitrogen fertilizer, we have to use the Haber-Bosch process, whereby we take nitrogen gas, we take hydrogen gas. It consumes a lot of energy. It consumes a lot of energy to make nitrogen fertilizer. But here you see these tiny soil microbes just doing it, not really effortlessly though. They use a lot of energy, but that tells us the superpowers of these microbes. So the process that we humans try to do with our batch process, trying to make nitrogen fertilizer, the rhizobia does it in plant roots. All they need to do is they have to form this mutualistic relationship with plants. Not all plants have that capacity, mostly with legume, like your soybean, cowpeas. The soil bacteria would attack and colonize the legume roots and then fix nitrogen. That's take that atmospheric nitrogen because it's in inert form, triple bond. It breaks it with a lot of energy and then make it available to the plants. And then in return, like I said, the plants will provide carbon for bacteria. And nitrogen fixation requires low or no oxygen. So that's why you see the plants to accommodate the rhizobia, they form nodules in their roots. You see those pinkish nodules here? It tells us there is an active nodule that is actively, whereby the rhizobia in there is actively fixing nitrogen. It's really beautiful to see the super capacity of these microbes. Not just nitrogen fertilizer alone, we have the phosphorus solubilizing microbes. Soils in tropical and subtropical regions, like you see the spodosaurs with altisaurs not so common in Florida. You see them, they're so reddish in nature, especially the altisaurs in this case. And that's because they're rich in iron, aluminum, and sometimes manganese. And these ions or cations in the soil will tie up phosphorus and make it unavailable most times to plants. But microbes have this capacity whereby they help to solubilize. If you have organic phosphorus not available, they help to mineralize phosphorus from organic phosphorus and solubilize phosphorus because phosphorus would attach to aluminum, iron, manganese, and making it unavailable to the plants. But the soil microbes will solubilize the phosphorus and make it available to the plants. So we call them phosphorus solubilizing microbes, increasing bioavailability of phosphorus for plants. And we have a wide range of bacteria and fungi that can do this. You see another superpower of soil microbes. And like I said earlier, soil microbes would also defend plants against pathogens. Bacterial colonization can induce plant responses to pathogens. You see them release volatiles, enzyme secondary metabolites that have antimicrobial properties. And also you see them releasing plant hormones, like I said, like ozins, gibberellins that helps to promote plants. You see the difference here, whereby this is a sun M where it has a lot of, you can see the difference between a plant that has been inoculated with microbes compared to the one that has not been inoculated. You see the difference in root growth. So that tells us the importance of soil. Sorry, there's noise here in the background. That's because of the screens being attached to the windows. I'm so sorry that disturbed. So we already said health is context dependent. You remember the slide, I presented it already. The parameters used for describing healthy soil in a temperate region is going to be different from the ones in a subtropical region like Florida. Now let's bring this on, talking about soil health and the importance of soil microbes. What are the common soil health challenges in Florida? This is the SCOTO soil, the national soil for Florida. And we can see, just looking at this figure, we can see how sandy it is. So most soils in Florida are very sandy. And what does that mean for our soil health? It means that these soils have low soil organic matter. It has poor soil structure, poor fertility. So what does that mean? It means that because the soil health challenges in our region is different from other regions, we have to carry out study, which is what we are doing right now, to look at the soil health indicators that we can use to measure soil health in Florida, because it's not going to be the same in other regions, because our challenges are unique. And now, you see this water body showing eutrophication. Environmental challenges are also tied to unhealthy soils. If the soil has poor structure, if it is very sandy, it means that whatever nutrients, if we apply nitrogen fertilizer with nitrate, it's going to be prone to leaching into water bodies. And apart from that, it means too that when we apply, let's say, phosphorus fertilizer, and there is a heavy storm or wind, we could have surface runoff too into water bodies. So it's not just, that's why the definition for soil was continued capacity of the soil to live as a living ecosystem, not just for plants, but for animals and we humans. So soil health is very important for everybody, because it's not just linked to plant production, but environmental challenges. I know when you go to the lake or you go to the beach, you want to have fun and relax in the water and be confident that the water you're going to for recreational purpose, it's safe. So that's why soil health is a topic that I feel like everybody should embrace. Now, we talked about the challenges of soil health in Florida, and one of them was low organic matter. Why is soil organic matter important to soil health? Now, on this figure, we can see crop residues, animal manures, they all help in building soil organic matter. In Florida, soil organic matter is a key to healthy soils in Florida. Why is that? And that is because soil organic matter helps in increasing nutrients and water holding capacity. It also helps to improve soil structure. And it serves as a carbon and energy source for soil microbes. We've talked about the different benefits of soil microbes. So it means that if we're going to increase these benefits that the soil microbes bring to the system, we have to increase the carbon and energy source for them. It's like you giving fish a bait when one goes fishing. Yeah, that was how it is too. Soil organic matter is very important. And what can we do to improve soil health and soil organic matter? We have different management practices. There are so many. I am just going to focus on these two because that's what we've been working on so far in our lab. And like I said, we have numerous practices that can improve soil health. And these different management practices, it kind of depends on the problems the challenges of soil health in the region. So that will determine the type of management practice to adopt. So I'm going to just focus on cover crops. What are cover crops? This here is a figure of a white mustard cover crop that was planted in 1929. So it means that cover crop is not new, it's something that has been there. But as in they adopted, I think recently there's an advent of adoption of cover crops, especially in citrus system, because of the HL, the citrus greening. So, what can we do to improve plant growth? What can we do? So that's where cover crops too comes into play. And cover crop is also known as a service crop because it's planted to benefit the soil and not just generally for profit. That's why it's called a service crop. It's it's becoming a common practice for grain, cotton, corn, soybean farmers, vegetable, and citrus farmers too. And the cover crop, it depends on your system. It depends on the system whereby this cover crop is being adopted. That will determine how it's being grown. In systems like row crop systems, cover crops is usually planted during the fallow season. But in citrus, because it's a perennial crop, cover crop is planted alongside with the crop. So it depends on your system. That would determine how the cover crop is being planted. And cover crop too is usually combined with conservation or no-tillage management practices. So there was a field trial a field trial in Southwest Florida, whereby they looked at the impact of cover crop on soil chemical properties and soil microbial communities. And they had different trees in different locations, and the cover crops were planted in that system twice per year, May and November. And the cover crops, for this study, which was conducted by Dr. Sarah Strauss and other lab members, they looked at different types of cover crops. The ones with legumes, where you had sun cowpea, and I know I talked about legumes too, forming relationship with soil microbes like rhizoma to fix nitrogen. And then they also have the non-legumes cover crop, the buckwheat, oats, rye, which you can plant at different seasons, summer, winter. And what did they find out? For this study, Castellano and Sarah Strauss, they looked at how different cover crop treatment affected soil organic matter and nitrogen. And the different bar colors represent the different treatment. The blue bar represents the controls in citrus system, the control where there was no cover crop planted. Then the green bar represents the non-legumes cover crop, and the red bar represents the cover crop where you add the legumes and non-legumes combined together. And they looked at the impact of these different cover crop treatment on soil organic matter and nitrogen in citrus system. Because I have different treatments here, there was no label on the x-axis, on the y-axis. So I'm looking at soil organic matter here, which is in percentage. You see that the place where they had cover crop planted, there was an increase in soil organic matter compared to the place where there was no cover crop. So that tells us the importance of cover crop on soil organic matter. And then they also looked at the different nitrogen, different nitrogens like nitrates. There was no effect of cover crop on nitrates. And then we look at the effect of cover crop on ammonia, which was in ppm, parts per million. You see that the place where we had cover crop, which is the green bar, the red bar, is greater than the citrus system where there was no cover crop. So that tells us now the importance of cover crop on soil organic matter and nitrogen. And if that is affecting soil organic matter and nitrogen, how does it affect soil microbial communities? You see this plot here. I know it's all so fancy, but the focus here is just looking at the different colors where we have green and red circles. Shows us those are the treatments where we have covered crop. And where you see blue, it shows you that this treatment was the control. And these different treatment, I'm looking at the bacteria now, it just shows the different time points. Where you have the square symbols, it just shows, at the start of the experiment. And after, I think 3 years after when cover crop was planted, you have the circle ones. You see that at the start of the experiment, we just have all the treatments together. That means there was no impact really on soil microbial communities. It's still the same thing for the fungi on the right-hand side. But now, when you see the circle, it shows us that these are the impacts of the cover crop after 3 years of planting. You see now, after 3 years, the control plot, see, there's no really significant impact on the soil bacterial community. But when you look at the green and red circles here, it tells us that after 3 years, the cover crop treatment actually influenced soil bacterial communities and soil fungi communities. That means there's really an impact of our cover crops planted. It actually impacts soil bacterial and fungal community composition. Now, this plot here, we call them ordination plots. They don't tell us the impact it's at. We can't really assess what impact, but it just shows us the impact. When you see this, you start going back to, okay, what type of bacterial and fungal communities were affected and what are their potential functions? So we also looked at, because now, remember when I said we look at what is there, who is there, what is there, and how they're related. Now, what is there? What do they do? This slide here just shows us nitrogen cycling genes. Start from nitrogen fixation. Well, for this, we looked at the total bacteria and total archaea. And you see that for the different treatments, when you look at these bars here, we have the cover crop where we have legumes and non-legumes. It's actually affected the gene copy numbers of the total bacteria and total archaea compared to the control plots. So it tells us that planting cover crop actually increased the abundance of bacteria and archaea. They are very important. But now we also looked at the specific genes that are involved in nitrification. What is nitrification? It's just conversion of ammonia into nitrous and nitrate. So, no, sorry, converts ammonia into nitrite and nitrate. And now we see that the ammonia oxidizing bacteria, these are the different microbes that are involved in this process: ammonia oxidizing bacteria and ammonia oxidizing archaea. So the blue bars shows us at the beginning of before cover crops were planted, there's really no impact of the cover crop treatment. Looking at the cover crop treatment on the x-axis, there's no impact of the cover crop treatment on the copy numbers or abundance of ammonia oxidizing bacteria and ammonia oxidizing archaea. But after 3 years, you see that the cover crops where we have legumes actually increases the abundance of ammonia oxidizing bacteria and ammonia oxidizing archaea compared to control and compared to the treatment with without legumes. So it tells us that cover crop actually affects these different nitrogen cycling genes and the total abundance of bacteria and archaea. But the type of cover crop also matters in this. So significant changes to denitrifying microbes with cover crop. What are denitrifying? These are the denitrification is the process of converting nitrite or nitrate into nitrous oxide, and also nitric oxide, nitrous oxide, and nitrogen gas. It shows that cover crop actually affected the denitrifying microbial. They're very abundant in soil, and bacteria, fungi, they are all responsible for denitrification in any agricultural system. So you see from this ordination plot, you see the colors of the different shapes here, triangle and circles. The green and the red shows us those are the cover crop treatment. And the blue square here is the no cover crop. You can see there's a distinction between the control plots and the ones with cover crop. So that tells us that when we introduce cover crop, it actually influences these different microbes that are responsible for denitrification, and like the previous slide, nitrification. For this, there's even a distinction too between the cover crop with legumes and cover crop without legumes. This is conversion of nitrous oxide to nitrogen gas. So even within the cover crop treatment, there's still a difference based on the type of cover crop, if it's legumes or non-legumes. Cover crop too also influences nitrous emission leaching. When we talk about nitrous greenhouse gases, you're talking about carbon dioxide, methane, nitrous emission. So you see that for this was a treatment whereby we had cover crop with legumes compared to non-legume cover crop and control. You can see that when we are combined cover crop, leguminous cover crop and non-leguminous cover crop, there was a decrease in the mean nitrous emission compared to the control plot. But meanwhile, for non-legumes, there was actually an increase. So it's not just about planting cover crop, but the type of cover crop to plant is also important. And that depends on, like we said, this context dependent. So this was a study that was conducted in North Florida, where they assessed cover crop in different cropping systems. They had the non-grazing integrated crop livestock system, sod-based rotation, where bahiagrass was integrated into row crop system. But let's just focus here on this figure, on these results in the red bar. You see, this bar shows us the difference between cover crop versus no cover crop and how it affects nitrate leaching. We can see here that where we have cover crop, there was a decrease in nitrate leaching compared to where there was no cover crop. So that tells us again the importance of cover crop. So what's the take-home message for this? Cover crops can change soil nutrients and microbiome composition and functions. Then the magnitude of changes can also depend on the cover crop mix, the legumes versus non-legumes. And cover crop can impact the environment's concerns related to nutrients, looking at nitrous oxide emission and nitrate leaching. What else can we do to improve soil health? I'll just go through this real fast. This compost, I think it was a plant-based compost that was added to citrus systems, and plant-based compost versus no compost. The compost was applied twice a year at 12.4 tons per hectare. And what do we see? Let's not focus on the colors here because it just represent different citrus rootstock. But let's focus on the shapes here where we have the squares versus the circle. What do we see? There is a difference in how there's a difference in the treatment with control. That's control at the square bars, and the circle, the ones with compost. So it tells us that when we apply compost to our system, it actually influences the soil microbial communities. You can see this here is the control versus the compost. And what else do we see? These are the gene copy number of bacteria. And then we see that the places where we applied compost, which is the red bar, the red box plot, they are higher compared to the blue where there was no compost. So that tells us that application of compost can affect abundance and also diversity of soil bacteria. So what's the take home for that? That compost can impact rhizosphere, soil microbes, depending on the rootstock. So what's the summary for this? Is that, like I said before, soil health is an analogy to help us think about ecosystem functions. Microbes are very important when it comes to soil health. Looking at their contribution, we looked at soil organic matter, nutrient cycling, and also protecting plants from diseases and pathogens. Cover crops and compost may impact soil microbes and soil nutrients in most agricultural systems in Florida. And because soil microbe is an indicator of soil health, is like the temperature of soil, monitoring soil health, we have to look at how it affects the soil microbes. So these are good organic management practices for soil health in citrus system in Florida. Yeah, these are the references. And with that, I will take any question. Thank you.
Thank you, Dr. Erhunmwunse, for a phenomenal presentation and webinar. I'm just going to take over here for the question. We have maybe 6 questions in the Q&A section. So the first one is coming from Alan Schumann. The question is,
Okay.
what is realistic percentage of organic matter in Florida lawns? So what is the realistic percentage that you're looking for when it comes to Florida lawns?
I would not say there is a realistic percent. All you want to see is an increase, right? It takes time for soil organic matter to really form compared to the way it's lost, it gets lost in the system. So instead of having that, creating a set, oh, this is a realistic amount, I think the focus would be, is it the whatever management practice that I'm using right now, applying to this system, is it increasing the soil organic matter? I think that should be the focus. For example, when I carried out my study, the soil organic matter was really low. I think it was 1% thereabout. And then I planted perennial legumes, and I saw an increase from 1% to 1.3%, which I was happy about because it shows me that there is an increase. So instead of saying a realistic number, I think I would rather focus on whatever I apply, am I seeing an increase? And it takes time,
Thank you, Dr. Erhunmwunse. The next question comes from Diane Goldberg, and the question reads, will the different types of soils affect the microbes and their ability to help the plants? So, and then they offer an example. So Rivera sand is acidic down to 28 inches, slightly alkaline, 28 to 42 inches, and moderately alkaline from 42 to 62 inches. So how do you determine what plants will do best in which environment?
Ask that question again. The last part of it, especially when you, yeah.
So the last part of the question is, how do you determine what plants will do their best?
Okay. I think so yes, the first part of the question, soil type, it actually affects, it actually would affect whatever management practice and how it would influence soil microbes. Yes, it would. My study, I talked about working with grass across Florida because I was looking at how integration of perennial legumes, the rhizoma peanuts, how it would affect soil microbes and soil health in general. And then we looked at different soil types across Florida. We looked at the 3 major soil types, the spodosol, altisol, and entisol. And I think apart from the soil type too, environment also played a role. So we found that the integration of perennial legumes, we saw a positive, I wouldn't say positive, let me not use the word positive, but we saw an increase in magnitude of how the rhizoma peanut affected soil microbes in some soil types, let me say altisols compared to entisols. And that's because of the soil types. There, it was really, really sandy. We did see impact of the rhizoma peanut on the soil microbes in entisols, but it wasn't compared to altisols. So yeah, the soil types would affect it too. And some of the soil types might take longer for you to see result compared to different soil types, whereby maybe after planting, let's say in this case, cover crop, it could take like a year or 2 or 3 before you see results. And maybe some other soil types, it could take longer than that before you see results. And apart from soil type, environment too would affect. Then the second part of the question was, can you please remind me then?
The second part of the question is, where would it be how do you determine what plants will thrive?
Oh, okay, perfect. How you determine what plants will thrive is just by doing research on it, or getting maybe speaking with professors that have done something similar in your area, And for instance, I think before I joined Dr. Sarah Strauss' lab, they've been looking at different types of cover crops that they can use to improve soil health in Southwest Florida. And what they did was do a first elimination study whereby they planted different types of cover crops just to see the one that did well. And from that result, they terminated, and then just looking at the data, or which one had greater biomass, which one increased the soil chemical properties, and then putting that data together. I think now it's still an ongoing study. We're going to use that data to determine which of these cover crops did better than others. So I think the best way to do that is just through experimentation.
Thank you, Dr.
Because
Erhunmwunse.
yeah, there are many factors involved.
So the next question is interested in some resources, maybe some EDIS publication or some fact sheets that will complement your presentation today. Do you have any of those resources available for the public?
I can always share with us after. I would have to pull those out. We do have some too that we just published. So yeah, I would have to pull those out and just compile them together and send to the organizers of this webinar, which they can send to everyone.
Thank you. I know we're going to take two more questions here. So the next one reads, are there particularly nutrient-rich cover crops that you would recommend that are resistant to snail and iguanas? Because those are the problems that they're having.
So snails and
Iguanas.
okay, I don't have any at the top of my head. I don't think going through all of the, I don't think right now that I do have anything at the top of my head. So this is a question that I will take to the team who have been working on cover crops for a long time, and then I can get back to us on that.
Thank you. And then the final question is coming from Diane Goldberg. It reads, would you recommend adding low-growing legumes like rabbit bells with grass to improve the soils or the overall health of the soil?
Yeah, it depends on the purpose of the legumes to improve the soil health. Then to why low growing? Could it be, what purpose is it used for? But I would always recommend legumes because you can see from our results that legumes actually increased the soil nitrogen and also reduced nitrous emission compared to just using non-legumes alone. And the good thing about legumes is it helps to form that symbiotic relationship with rhizobia. So nitrogen fixation is ongoing, which is really good too for the soil microbes and in general soil health. So yeah, I would always recommend legumes to be integrated with non-legumes and just to help improve and get the benefit from both sides. And then also reduce maybe input of nitrogen fertilizers. But
Thank
yeah,
you for
like I
that
said, it
response.
actually depends on the purpose.
Well, with that takes us to the end of our question list. Dr. Erhunmwunse, if you don't mind sharing with everyone where they can keep up with your future research, with some of the collaboration that you're coming on, that you are ongoing with, in case someone has a question or would like to get in contact with you after this webinar, please advise how they can get in contact.
Yes, so if you have questions and you want to keep up with what we're doing, you can always reach me on my email, which is on the, I don't know if you all can still see my screen now. I will just spell that out, ERHUNMWUNSEA. You can always reach me via email. And you can also reach the PI of our lab, Sarah Strauss. And our email is strauss@t. I'll just type that now. So that way, if you have questions, yes. The PI of our lab, Dr. Sarah Strauss, you can also reach her, strauss@ufl.edu, and we'll be more than happy to respond to your questions. And like I said, after this, this webinar, I'm gonna put together the resources and share to the organizer of the webinar, and then share with everyone for more information on soil health.
Thank you, Dr. Erhunmwunse, and thank you everyone for your time for this webinar today. This is going to be the end of the webinar. And I do apologize if I didn't get a chance to cover all of the questions answered, which is why we now have the contact information of Dr. Erhunmwunse, right? So thank you again, and everyone all take care.
Thank you.